Imagine for a moment you're trying to protect something incredibly precious, the absolute core of who you are. Your whole existence relies on keeping it safe, totally sealed off from, well, the chaos and dangers outside. What would you do? You'd build the most secure, most impenetrable fortress possible, right? Well, guess what? Your brain already has one. It's this amazing biological fortress, silently guarding your most vital organ every single second. Today, we're doing a deep dive in this incredible guardian, the blood-brain barrier, the BBB. Sounds simple enough, maybe, but its importance for how our brain works, stays healthy, and even fights off disease. It's just profound. Our mission today is to give you a shortcut, really, to understanding this critical interface. We'll look at how it was found, how it works, all its intricate parts, and why it's such a hot topic in neuroscience right now. Okay, let's unpack this ultimate protector.
Yeah, let's do that. So at its heart, the blood-brain barrier, the BBB, it's this highly selective, semi-permeable boundary. Think of it like a microscopic filter, a really sophisticated one. It's formed by these specialized cells, endothelial cells, that line the tiny blood vessels, the capillaries, inside your brain. But what makes them really special are these things called tight junctions between the cells. They're incredibly tight, essentially zipped together, forming a physical and biochemical shield between your brain and spinal cord, your central nervous system, and the blood flowing through the rest of your body.
Wow. So it really is like the ultimate bouncer for the brain, standing guard, blocking harmful chemicals, germs, toxins that might be floating around in our blood. But, and this is the clever part, it's not just about keeping things out. It also lets in the good stuff.
Exactly. It's a brilliant strategist. It selectively allows vital nutrients, things like glucose, which is the brain's main fuel, specific amino acids, certain ions, things the brain absolutely needs to function. It makes sure the brain operates in its own stable, protected little world, free from disruptions.
That's key. And what I find fascinating is how we even figured this out. It wasn't just one discovery, was it?
No, not at all. What's truly fascinating here is how this understanding built up over time. It wasn't instant. It was piece by piece, like detective work, challenging earlier ideas.
Right. Here's where the story gets really interesting. Because, yeah, the discovery was kind of roundabout. It started back in 1885 with a German scientist, Paul Ehrlich. He was doing experiments injecting dyes into animals. And he noticed something odd. The dyes stained pretty much every organ in the body. Liver, kidneys, heart, you name it. Everything except the brain and spinal cord, they just stayed clean. His first thought, he figured the brain just didn't have much affinity for the dye, like it didn't soak it up well, a property of the brain itself.
Right, that was the initial assumption. But then, almost 30 years later, in 1913, one of Ehrlich's own students, Edwin Goldman, decided to test that idea further. Goldman tried a different approach. He injected the dye directly into the cerebrospinal fluid, the fluid surrounding the brain.
Ah, okay. Inside the barrier, basically.
Exactly. And this time, the result was the complete opposite. The brain did stain, but the rest of the body didn't. That was a huge piece of evidence. It strongly suggested there wasn't just low affinity, but an active selective barrier doing the separating. It showed Ehrlich's first idea wasn't quite right.
And then came Lena Stern, right? She put a name to it.
Yes, Lena Stern, a brilliant Russian neuroscientist around 1921. She's the one who actually coined the term hematoencephalic barrier, literally blood-brain barrier. But she did more than just name it. She really advanced the understanding of its physiological role. She emphasized that it wasn't just a passive wall-blocking things, it was dynamic, a functional interface that actively regulates what gets across, an active gatekeeper. Not just a fence.
Okay, so that really set the stage for how we see it now.
Precisely. And if we connect those early observations to what we know today, well, the BBB isn't just a single wall. It's incredibly complex, a multicellular system. Think of it like that fortress again, but built with many layers, different specialists working together. Each part has a distinct job, but they all cooperate to protect the brain's very delicate environment. It's remarkably precise.
A whole team of specialized cells, not just one type. That sounds like, yeah, amazing biological engineering.
It truly is. So first up, you have the endothelial cells. They're the main gatekeepers, forming the actual lining of those brain capillaries. Now, unlike capillaries elsewhere, these cells are stitched together with those super tight junctions we mentioned. Practically nothing can squeeze between them. That stops stuff just leaking through. They also have a very low penocytotic activity. That means they don't just gulp down fluids and substances randomly like other cells might.
Much more controlled.
Very controlled. Instead, they use highly selective protein transporters embedded in their membranes. Think of them like specific doorways. GLUT1 for glucose, the brain's fuel. LET1 for essential amino acids, building blocks for proteins and neurotransmitters. They actively ferry these crucial molecules across.
Got it. So endothelial cells are the front line, the main wall with specific doors. Who else is on the team?
Okay, next layer, wrapping around these endothelial cells are the pericytes. Think of them as regulators, almost like support staff managing the wall. They're really critical for barrier integrity. They can actually contract or relax, which helps control the diameter of the capillaries. That fine-tunes blood flow to different brain areas.
Wow.
Yeah, and they directly support the endothelial cells, helping them form and maintain those vital tight junctions. They're also involved in repair if the barrier gets damaged, and they help control how much stuff gets transported through the cells themselves, keeping it limited.
And I think I've heard about astrocytes, those star-shaped cells.
Yes. The astrocyte enphyte, these are glial cells, a type of support cell in the brain. They have these distinctive star shapes and send out projections called enphyte, and these enphyte densely surround almost the entire surface of the brain capillaries, maybe 98% coverage. It's incredible.
Oh.
They're not just passive insulation either. They actively signal to the endothelial cells, releasing chemicals that help maintain those tight junctions.
Oh.
So the endothelial cells, the pericytes, and these astrocyte enphyte, they all work together as this integrated system. We call it the neurovascular unit. It keeps the barrier strong and the brain's internal environment stable.
That's just an amazing level of coordination, like a tiny biological symphony. Is there anything holding it all together structurally?
There is indeed the basement membrane. This is a thin layer, sort of like a specialized mesh or scaffold right outside the endothelial cells. Think of it as the strong, flexible foundation the fortress wall is built on. It provides mechanical support, physically separates the blood vessel from the brain tissue, and importantly, it also acts as a signaling platform, helping all these different cell types communicate with each other. It's made of tough proteins like collagen and laminin.
Okay, so we have this incredible multi-layered structure all working together. Beyond just being a physical barrier, what are its main day-to-day jobs? Protection must be number one.
Absolutely. Protection is the primary function. It shields the brain from harmful substances circulating in the blood, bacteria, viruses, toxins, even large molecules that shouldn't be there. This massively reduces the risk of brain infections and protects neurons from potentially damaging chemicals. It's the fortress wall doing its job.
And keeping things stable inside, maintaining balance.
Exactly. That's homeostasis. The BBB constantly works to stabilize the brain's internal environment. It carefully controls the levels of ions, like potassium and calcium, which are critical for nerve firing. It manages neurotransmitter levels, keeps the pH balanced. Without this tight regulation, the delicate electrical activity of neurons would be totally disrupted. Think seizures, cognitive problems. It maintains that perfect internal climate.
Does it play a role in immunity too? Like
Yeah.
deciding which immune cells get in?
Definitely. It acts as an immunological barrier. It strictly limits the entry of immune cells from your bloodstream T cells, B cells, macrophages into the brain. This is crucial to prevent unwanted inflammation and autoimmune attacks on brain tissue, which can be devastating like in MS.
Right.
But it's not a complete lockdown. If there's a serious infection, for instance, the BBB can allow controlled specific entry of certain immune cells to help fight it off. It's regulated surveillance.
How does it manage to be so selective? Letting the good stuff in, keeping the bad stuff out. That seems like the real trick.
It is the trick, and it uses several clever selective transport mechanisms. First, there's passive diffusion. This is for small, fat-soluble molecules, things like oxygen, carbon dioxide, alcohol, nicotine. They can just slip right through the cell membranes easily following their concentration gradient. No special transporters needed.
Okay, simple enough for those.
Then, for essential molecules that aren't fat-soluble, like glucose and amino acids, the brain uses carrier-mediated transport. These are specialized proteins acting like dedicated shuttles or specific doors. They bind to the molecule and carry it across the barrier, like bouncers checking a guest list. GLUT1 for glucose, LET1 for amino acids, as we mentioned.
Got it. Shuttles for the essentials. What about bigger things like hormones?
For larger molecules, things like insulin or transferrin, which carries iron, there's receptor-mediated transcytosis. These molecules bind to specific receptors on the endothelial cell surface. The cell then kind of engulfs them in a tiny bubble, a vesicle, and transports them across to the other side. It's like a special VIP delivery service.
Okay, that's clever.
And finally, there's an important defense mechanism: efflux pumps. Proteins like P-glycoprotein actively pump unwanted substances out of the endothelial cells, either back into the blood or preventing them from fully entering the brain in the first place. They're like security guards ejecting potential troublemakers or drugs the brain doesn't want.
And it even detoxifies things before they cross. That's amazing.
It does. It also functions as a metabolic barrier. The endothelial cells themselves contain enzymes that can break down or modify certain molecules, including some drugs or toxins, neutralizing them before they even get a chance to reach the brain tissue. So yeah, if you connect all these pieces, the blood-brain barrier is this incredibly dynamic, cooperative system. Every part has a job, all synchronized to protect the brain's unique and very sensitive environment.
Okay, so this fortress is incredibly tight, incredibly specific about what gets in and what stays out. Genius engineering. But you mentioned earlier, there are places where it's intentionally not complete, these circumventricular organs, CVOs. Why would the brain have deliberate weak spots?
It sounds counterintuitive, right? But it's actually a brilliant adaptive design. These CVOs are specific little regions, mostly located around the brain's fluid-filled ventricles, where the barrier is leaky on purpose. They need direct access to the bloodstream to monitor what's happening in the body. They sense hormones, nutrients, potential toxins, osmotic pressure, things circulating in the blood.
Ah, so they're like listening posts.
Exactly. They allow the brain to rapidly detect changes happening systemically and then trigger appropriate responses, maybe releasing hormones, making you feel thirsty, or even inducing vomiting if you've ingested something toxic. They help coordinate the brain with the rest of the body. We can group them into sensory and secretory types.
Okay, tell me about the sensory ones first. What are they sensing?
The sensory CVOs are all about detection and relaying information. A classic example is the area postrema. It sits in the brainstem and is often called the vomiting center. It directly samples the blood for toxins, certain drugs, or hormones that signal trouble, and then triggers the nausea and vomiting reflex to get rid of harmful substances. That's why chemotherapy can make you feel sick, for example.
That makes total sense, a built-in defense mechanism. What else are these sensory CVOs watching?
Well, there's the subfornical organ, the SFO. Think of it as your fluid balance sensor. It's very sensitive to hormones like angiotensin II, which signals low blood pressure or dehydration. The SFO detects this and then triggers feelings of thirst and stimulates the release of hormones to conserve water. It helps manage your body's fluid balance.
Okay.
Then there's the organum vasculosum of the lamina terminalis, or OVLT. This one detects changes in blood saltiness, osmolality, and also senses substances called pyrogens, which are released during infection and cause fever. So the OVLT helps regulate thirst and body temperature, initiating fever responses when needed.
Right, so these are like critical internal monitors. And then the secretory CVOs, they're designed to release things directly into the blood, bypassing the barrier.
Precisely. These areas specialize in producing neurohormones or other signaling molecules and releasing them straight into the general circulation so they can travel throughout the body. A good example is the pineal gland. It's a secretory CVO that produces and releases melatonin, the sleep hormone, directly into the bloodstream to regulate our daily sleep-wake cycles, our circadian rhythms.
Right.
Another crucial one is the median eminence. This acts as the key link between the hypothalamus in the brain and the pituitary gland. Hypothalamic releasing hormones travel through the median eminence to enter the pituitary's blood supply, controlling the release of hormones that regulate growth, reproduction, stress, metabolism, basically coordinating the brain's control over the body's endocrine system. And the neurohypophysis, or the posterior pituitary gland, is another. It doesn't make hormones, but it stores and releases vital ones like oxytocin and vasopressin, also called ADH, directly into the blood. Oxytocin is involved in things like childbirth and bonding, while vasopressin regulates water balance. There's also the subcommissural organ, or SCO, which secretes glycoproteins into the cerebrospinal fluid, thought to be involved in CSF flow and maybe neurodevelopment.
So these aren't flaws at all. They're incredibly smart adaptations. Strategic openings allowing the brain to monitor and talk back to the body directly. Truly remarkable.
Yeah.
But this brings us back to the danger zone. What happens when that main fortress wall, the BBB, gets breached when it's not supposed to be, when it's damaged?
Yeah, and this raises an important and quite serious point. If the BBB is compromised unintentionally, it flips from being a protector to becoming a pathway for harm. It can actually worsen the onset and severity of diseases. When the barrier breaks down, harmful substances, inflammatory cells, pathogens can flood into the brain's delicate environment. This leads to neuroinflammation, direct damage to neurons, and contributes to a whole range of neurological disorders.
What kind of things cause that damage? What breaks the barrier down?
Several culprits. Oxidative stress damage from unstable molecules called free radicals is a big one. Inflammation itself is key. Inflammatory signals, called cytokines, can directly attack and loosen those tight junctions between the endothelial cells. Infections, obviously, where bacteria or viruses actively target or weaken the barrier. High blood pressure puts physical stress on the vessel walls, making them leaky. And exposure to certain environmental toxins can also directly poison the barrier cells. The result is always increased permeability, leakage, which fuels inflammation and neuronal damage, making brain diseases worse.
Can you give some examples? What specific diseases are linked to BBB breakdown?
Certainly. Let's take Alzheimer's disease. We see impaired clearance of amyloid beta peptides across the BBB. The transporters aren't working right. And then the amyloid beta that builds up actually further damages the barrier itself. It becomes a vicious cycle.
Oh, wow.
In Parkinson's disease, inflammation and problems with the cell's energy factories, the mitochondria, contribute to BBB breakdown. This allows inflammatory factors in and might disrupt the transport systems needed by the dopamine-producing neurons that die off in Parkinson's.
And something like multiple sclerosis, that involves the immune system attacking the brain, right?
Exactly. In MS, it's thought that initial inflammatory triggers compromise the BBB. The barrier becomes leaky. This allows those peripheral immune cells, which are normally kept out, to cross into the brain and spinal cord where they mistakenly attack the myelin sheath, the protective coating on nerve fibers. That leads to the characteristic damage and symptoms of MS.
And what about sudden events like a stroke or head injury?
Big impacts there, too. In stroke, the lack of blood flow causes damage, triggering a massive inflammatory response. Same with traumatic brain injury, TBI. The physical trauma sets off inflammation. In both cases, inflammatory molecules rapidly break down the tight junctions. The barrier becomes highly permeable, leading to swelling in the brain, edema, further cell death, and long-term neurological problems.
And infections getting through. You mentioned pathogens are clever.
They are. In bacterial meningitis, for example, bacteria directly invade and inflame the meninges, the layers surrounding the brain, often compromising the nearby BBB. And HIV, the virus that causes AIDS, can use a particularly insidious method. It infects immune cells called monocytes, and then essentially uses them as a Trojan horse to get carried across the BBB into the brain, leading to neurological complications.
Okay, it's incredibly clear how vital a healthy BBB is and how damaging a breach can be, which leads directly to a huge challenge in medicine, right? Drug delivery. If this barrier is so good at keeping things out, how on earth do we get treatments in for all these brain conditions?
It's arguably one of the biggest hurdles in developing treatments for brain diseases. The numbers are stark. Something like 98% of potential small molecule drugs and virtually all large molecule drugs, like antibodies or proteins, simply cannot cross the BBB on their own. This massively limits our options for treating brain tumors, Alzheimer's, Parkinson's, epilepsy, infections. You name it, it's a formidable obstacle.
But researchers are finding ways around it, aren't they? It's incredible how creative they're getting to sneak medications past this shield. What are some of the strategies?
They really are getting creative. One major approach is using nanoparticle-based delivery. The idea is to encapsulate the drug inside tiny nanoparticles like little fatty bubbles called liposomes, or microscopic polymer spheres. These can be designed to be small enough or have specific surface properties to help them cross the barrier.
Like tiny delivery trucks.
Exactly. And sometimes they add ligands like specific antibodies to the surface of the nanoparticle. These ligands act like keys, binding to receptors on the BBB cells and tricking the barrier into actively transporting the nanoparticle across. It's often called a Trojan horse approach. It helps target the drug delivery too.
Okay, disguising the cargo. What about changing the drug itself?
That's another key strategy. Chemical modification of the drug. Scientists might tweak the drug structure to make it more lipophilic, more fat-soluble, so it can diffuse more easily through the cell membranes of the BBB, or they might temporarily mask parts of the drug molecule that prevent it from crossing. Sometimes they create prodrugs, inactive forms of the drug that can cross the barrier and are then converted into the active drug once inside the brain.
And I heard about using ultrasound. That sounds pretty sci-fi.
It does, doesn't it? It's called focused ultrasound, or FUS. It's a really exciting, non-invasive technique. Doctors inject tiny microbubbles into the bloodstream, then use highly focused ultrasound waves aimed at a specific brain region. The ultrasound makes the microbubbles oscillate, and this temporarily and safely opens the tight junctions of the BBB just in that targeted spot.
Just for a short time.
Yes, just temporarily, allowing drugs that are also circulating in the blood to enter that specific area. It's still largely experimental, but showing huge promise in trials for getting chemotherapy to brain tumors or delivering therapies for Alzheimer's with much less systemic exposure. Researchers are also trying to hijack the natural receptor-mediated transport systems. If they can design a drug or attach it to a molecule that naturally binds to receptors like the insulin or transferrin receptor on the BBB, they can potentially get it shuttled across using the brain's own delivery mechanisms.
And of course, some drugs just naturally get through. Yeah.
Yes, that's true. We rely on that for many existing brain medications. Certain drugs are already inherently lipophilic enough to cross the BBB via passive diffusion. This includes many common psychiatric medications like antidepressants, antipsychotics, and anti-anxiety drugs like benzodiazepines. Their chemical nature allows them to slip through the barrier relatively easily.
Okay, so we've got this amazing external fortress, the BBB. But the brain also has its own internal security force, right? The microglia. How do they fit into this picture?
Microglia are fascinating. They're the brain's resident immune cells, kind of like macrophages, but specialized for the central nervous system. And they have this really interesting dual role when it comes to the BBB. In a healthy brain, they're protective. They patrol the environment, clean up cellular debris, look for signs of trouble. And importantly, they release factors that actually support BBB integrity, helping to maintain those tight junctions and keep the barrier strong. They're guardians.
So normally they're helpful janitors and guards, but I get the sense they can also cause problems.
They absolutely can. When there's injury, infection, or chronic inflammation, like in Alzheimer's or Parkinson's microglia, can become activated in a harmful way. They shift into what's called a pro-inflammatory, or M1 state. In this M1 state, they pump out a flood of inflammatory molecules, cytokines like TNF-alpha, IL-1 beta, and reactive oxygen species, which cause damage. Crucially, they also release enzymes called MMPs, matrix metalloproteinases. These enzymes can directly chew up the proteins forming the tight junctions, like claudin-5 and occludin.
So they actively break down the barrier.
Yes, they directly contribute to BBB breakdown, increased leakiness, and neuroinflammation. It exacerbates the disease process.
Wow. So these tiny cells are definitely a double-edged sword. They can protect the fortress or help tear it down.
Exactly. But there's hope on the other side of that coin. Microglia can also shift into an anti-inflammatory, or M2 state. This M2 state is associated with tissue repair and resolving inflammation. M2 microglia release anti-inflammatory signals, help clear up debris without causing more damage, and actually promote the repair of tight junctions and the survival of endothelial cells.
Ah, so switching them from bad guys back to good guys.
That's a major therapeutic goal now. Finding ways to encourage microglia to shift from that damaging M1 state towards the healing M2 state is a really hot area of research for conditions like Alzheimer's, stroke recovery, and MS.
This is all just incredible, the complexity protecting our brains and how even the internal defenses can play both sides. It makes you wonder, what can we do? Can our everyday habits actually influence something as complex as the blood-brain barrier?
Absolutely. And this is maybe the most empowering part of the story. The health and integrity of your BBB are directly influenced by your daily lifestyle choices, which really raises an important question for you listening right now. How can your choices become active strategies to help preserve this vital barrier for your own brain health?
Okay, give us the practical advice. What works?
Well, first off, an anti-inflammatory diet is really foundational. Focus on foods rich in antioxidants. Think colorful fruits and vegetables like berries, spinach, kale, beets, broccoli. They help combat oxidative stress that damages barrier cells. Spices like turmeric, thanks to its active compound curcumin, has powerful anti-inflammatory effects. And omega-3 fatty acids are crucial. Find them in fatty fish like salmon, flax seeds, chia seeds, walnuts. They support healthy cell membranes and those all-important tight junctions.
And what should we avoid?
Definitely try to minimize processed foods, refined sugars, and unhealthy trans fats. These tend to promote systemic inflammation, which we know can directly harm the BBB.
Makes sense. What about exercise? We hear it's good for the brain generally. Does it help the BBB specifically?
It absolutely does. Regular physical activity provides multiple benefits for the BBB. It improves blood flow to the brain, ensuring good oxygen and nutrient delivery. Exercise also helps lower systemic inflammation throughout the body. Plus, it promotes the growth of new neurons and encourages healthy blood vessel structure, all of which supports a strong barrier.
And sleep. How important is that?
Sleep is critical. During deep sleep, the brain activates its unique waste clearance system, the glymphatic system. This system works kind of like a power wash, flushing out metabolic waste products, including things like amyloid beta, which can damage the BBB if they accumulate.
So lack of sleep is bad news for the barrier.
Yes, studies show that chronic sleep deprivation can actually increase BBB permeability, potentially by disrupting tight junction proteins and promoting inflammation. Aiming for that consistent 7 to 9 hours of quality sleep is really important for barrier maintenance.
What about other common health issues like high blood pressure or blood sugar?
Huge impact. Both hypertension, high blood pressure, and uncontrolled high blood sugar, hyperglycemia, often seen in diabetes, are major threats to the BBB. High pressure puts physical strain on the delicate endothelial cells, while high sugar levels cause oxidative stress and inflammation, damaging the cells and loosening the tight junctions. So, managing blood pressure and blood sugar through diet, exercise, medication if necessary is absolutely vital for protecting your BBB.
Okay. And are there any bad habits we should definitely ditch for our BBB's sake?
Yes. Excessive alcohol intake can directly impair tight junctions and increase permeability, plus it promotes damaging oxidative stress over time. Smoking is another big one. The thousands of toxins in cigarette smoke generate free radicals and trigger inflammation, both of which directly harm the BBB.
Right.
And maybe one people don't think about as much, chronic stress. Constantly elevated stress hormones like cortisol can weaken the BBB and also push those microglia towards that inflammatory M1 state we talked about, further damaging the barrier. Finding healthy ways to manage stress, mindfulness, exercise, yoga, whatever works for you is really beneficial.
So it's really empowering to know our daily choices matter so much for this hidden protector. It's not just out of our hands. Okay, looking forward now, where is the research heading? What's the future for the blood-brain barrier?
Well, if we look at the big picture, the BBB is really shifting focus in research. It's moving from being seen primarily as just an obstacle we need to overcome for drug delivery to being understood as a dynamic player in disease itself, and potentially a therapeutic target or even a gateway for new treatments. It's a major frontier in neuroscience and medicine right now, holding huge promise.
What are scientists actively working on? What are the hot topics?
There's a lot of focus on BBB permeability studies. Researchers are trying to understand precisely why and how the barrier becomes leaky in different diseases, stroke, Alzheimer's, MS, infections. They're zooming in on those tight junction proteins and how they're regulated. We're also seeing incredible advances in imaging techniques, things like specialized MRI, PET scans. They're allowing us to actually visualize BBB leakiness in living brains, sometimes even track drugs crossing the barrier in real time. That's huge for research and diagnosis.
And testing things in the lab.
Yes, there's amazing progress in vitro and in vivo models. Scientists are creating sophisticated organ-on-a-chip devices and 3D spheroid cultures that mimic the human BBB much more accurately than older methods. This speeds up drug testing and helps us model diseases better.
And beyond just getting drugs across, are there other therapeutic ideas being tested?
Absolutely. Gene therapy is being explored, using modified viruses like AAV vectors to deliver corrective genes directly into brain cells, potentially bypassing the need to cross the BBB with a drug. Stem cell therapy is another exciting area. Research is looking into whether stem cells can help repair a damaged BBB or release protective factors to restore its integrity. There are early trials ongoing for conditions like stroke and TBI.
Is it possible to tell if someone's BBB is compromised, maybe before they have major symptoms?
That's a critical goal, developing reliable biomarkers of BBB integrity. Researchers are hunting for specific molecules in the blood or cerebrospinal fluid that indicate the barrier is leaky. Things like the ratio of albumin in CSF versus blood, or levels of proteins like S100B or neurofilament light chain, NFL, or those MMP enzymes we mentioned. Having good biomarkers would be huge for early diagnosis and for tracking whether treatments are working.
So looking even further ahead, what might be next? What's the dream?
The horizon is really exciting. We might see things like CRISPR gene editing being used very precisely to fix genes related to BBB function or dysfunction. We'll likely see more development of BBB-targeted neuroprotective drugs therapies designed not just to get through the barrier, but to actively strengthen it, maybe by boosting tight junctions or calming those inflammatory microglia. And ultimately, the dream is moving towards personalized BBB-based therapies, where treatments could be tailored based on an individual's specific BBB characteristics and their particular disease.
Wow. This is incredible. From being discovered almost accidentally with dyes to becoming this central player in brain health and disease, the blood-brain barrier is truly remarkable. It's not just a wall. It's this living, dynamic, intelligent system that protects your brain, manages its environment, and connects it to your body. So what does all this mean for you? Well, it means understanding the BBB isn't just some academic exercise. It's absolutely key for pushing forward treatments for devastating neurological disorders. And, maybe just as importantly, it empowers you, knowing that your lifestyle choices can genuinely support your brain's ultimate protector. Considering everything we've explored about this amazing vigilant barrier, it really makes you wonder, doesn't it? What incredible new insights will unlocking its secrets reveal next in our quest for better brain health? It means you're now one step closer to being truly well-informed about your brain's ultimate protector.